Respected dignitaries, Principal Ma'am, Dr. Anuradha M., dear colleagues, and my beloved students, a very good morning to one and all. As we all know, science is any stream of knowledge that is concerned with the physical world and its phenomena, and that entails unbiased observations and systematic experimentation. In general, a science involves a pursuit of knowledge covering general truths or the operations of fundamental laws. Sciences around us, the process of creating the air we breathe, also science. The food we enjoy, water we drink, and clothes we wear are all based in science. Looking up into the atmosphere gives us a glimpse into astronomy, another branch of science. You can't get around it. Science is everywhere and is one of the most important topics of study in our world. Science can be divided into different branches based on the subject of study. The physical sciences study the inorganic world and comprise the fields of astronomy, physics, chemistry, the earth sciences. The biological sciences, such as biology and medicine, study the organic world of life and its processes. Social sciences, like anthropology and economics, study the social and cultural aspects of human behavior. Sciences further treated in number of articles. So to begin with, let's start the event with an invocation song. Let's join our hands for the prayer. Akshay sir.
आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपो हरतु मे पापम् सन्ध्यादीपनमोऽस्तु ते ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः आरोग्यं देहि पुत्रांश्च मतिं स्वच्छां हि मे सदा रवेरस्तं समारभ्य यावत् सूर्योदयो भवेत् रवेरस्तं समारभ्य यावत् सूर्योदयो भवेत् यस्य तिष्ठेत् गृहे दीपः तस्य नास्ति दरिद्रता शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः
In science context, Science Forum platform in our institutions gives an opportunity to one and all to celebrate science in any form. We can enhance and exchange our knowledge through presentations, discussions, skits, songs, dances, mimes, and so on. It's a great opportunity for all of us to utilize the platform and enlighten our knowledge. Our principal ma'am is always a great support for these scientific activities. To have more insightfulness, now I request our beloved principal ma'am, Dr. Anuradha M., today's guest of honor, to give inaugural address. It's always my privilege and immense pleasure to introduce her to the gathering on behalf of Padma Shri Institute of Management and Sciences. Ma'am has completed her PhD in Krishnadevaraya University, Anandpur. She started her career as a scientist and lab in charge in Dr. Reddy's labs. She worked as a senior scientist and head in semi labs Bangalore. Later, she started her own company, Rishi Herbal Technologies Private Limited, in the year 2004. She joined Padma Shri as a principal in the year 2004. She is also the director of PIMS Research Center and Biotech Finishing School from 2011 to till date. She has the academic and research experience in nutraceuticals, medicinal plants research, and plant biotechnology. She worked with national and international agencies for developing novel nutraceutical products for human and veterinary use. She is the BOS chairman and BOE at Karnataka Open University. She is also BOE member in JSS College for Women Mysuru and MLA CW Bengaluru and BOS member at Dayanand Sagar University. She is the member of the Academic Council, Bangalore University. She has organized many seminars and conferences at the national and international levels, participated and presented various research papers at the international, national, state level seminars, conferences, and workshops. And also, Man has attended many national seminars as a resource person. She has filed patents for many of her research, and one was US patent granted. She has completed many projects and ongoing projects funded by DST, KBITS, Government of Karnataka, Department of ITBT, and ST Government of Karnataka. She published many of her research papers in reputed journals. She is an eminent personality with great research knowledge. She is proactive and guide all of us to enhance our scientific knowledge through research work. She always encourage and support us for the scientific work, and Science Forum is one amongst them. We are fortunate to work under her guidance. A hearty welcome to you, ma'am. Over to you now.
Thank you, Netra, for your kind and elaborate introduction, which was actually not required because all of you know about me. And see, this Science Forum platform is being initiated long back in our institution with a small initiative to make all of you to come onto one platform, because most of you will be pursuing your own interested career, and most of you may not be getting in touch with each other to share your scientific knowledge or to share your experiences. Science is I don't want to define science and tell how it is integrated in our lives. It is part and parcel of our life, and especially the year 2020 can be called as year of scientists. It is a year where entire world is looking towards the scientists and biologists. This is the year where we understood the importance of life science. This is the year we understood the importance of medicine and biological sciences. This is the year where we really realized that we cannot separate science from us. It is our integral part of our life, and it taught us many things which we can never learn in our life, even if you get your PhD or postdoctorate or 10 doctorates also. That's what the importance of life, importance of science, importance of every being in this universe is being taught to us. A small, tiny RNA-based virus could able to rule the world for past 9 months, and still it is doing so. With this small thing, I want to tell you what we do in this science forum. Most of you may be having the curiosity, what does it mean, the science forum? The science forum is a platform for all of you to open up, to give your ideas, to conduct various, it can be a scientific song, it can be a poem, it can be a drawing, it can be a skit, which really showcases the science, ancient science or modern science or the current science, whatever is happening around us. Let us try to take this science forum to reach masses, where we can have certain collaborations or certain adoptions. You can adapt some schools nearby whenever you can, when anyhow on one day we are going to see each other offline, which is not going to be any more virtual meetings. At that time, we can definitely adopt one of the village, and you can go and teach them the science. Actually, there are many national international organizations which are giving some kind of funds to take the science to masses, where you can go and really help them out. For example, DST had Rajat Jayanti Vignan Sancharak fellowships. Now it is not there in DST, but some other organization surely will have, that is called as science communicators. These science communicators, the responsibility of science communicators is you have to go and teach them. We got some projects in this science communicating area under DST. Now also there are certain small research fundings opened up in SSP. I request all of you to submit your projects for this, and your teachers might be having the format and everything is there. If still last date is there, please try to apply for this. That will ignite you, encourage you to do certain small things. What do you mean by small things? For example, you might be having a water bottle, a water can lying waste at your house. You can create a hydroponic system, and you can borrow certain saline bottle tubes and connect it so that you can have drip irrigation in your house. And you can have gardens, to have gardens in small place also and with less water. This kind of many interesting ideas you might be getting, and these ideas can be shared here, and you can create, we are creating a platform, and slowly we can take it up to the next level. I'm happy to say that here today they are inaugurating a journal, a scientific magazine from Padma Shri Institute of Management and Sciences, rather Padma Shri group of institutions. I wish all of you can share your knowledge and write articles and whatever. It may be creative version of science, it may be anything which is related to science. You please share your articles so that they can be published. Initially, we are doing e-journal, that is only soft copy of this will be distributed to all of you. And later we may go for the print media also if everything goes right. I congratulate the organizers, Netra and Sunita and all the team members of Padma Shri Institute of Management and Sciences, who are working back end and who are working in the front end to make this science forum a successful initiative. This being the first session of science forum, I thought I will give a small brief introduction, so I took little time. But anyway, that is required for all the newcomers, especially who joined this year. We have first year UG people, BSc, BSc CND, BVOC, and BCA. And also we have a BICEP online certification course program students also in this science forum. I welcome all of you for this science forum. Please go ahead. Netra, over to you, Netra.
Thank you very much, ma'am, for addressing the gathering. And thank you so much for your valuable and motivating words. Literally thankful to you, ma'am. Now it's over to Shalini and Swati for further Science Forum activities for today.
Thank you, ma'am. I would like to thank Anuradha ma'am for the valuable and motivating words, and also telling briefly what the science forum tells like means. So as ma'am said, science forum is a platform where like anyone is open to share their views or interests on any of their topics in any means. So for here we have few participants to uh, present on the topic. So this forum is basically set up because there's a thought that a secret of getting ahead is getting started. So the reason for starting the science forum is to gain more knowledge and to gain more interactive sessions and all, and to have like to spread the knowledge of science. So over to Swati.
Now, let's start this science forum with a skit. Um, there is a skit presented on the global warming greenhouse effect by Ranjini, Riya, Sadhana, Shravan, and Spurti from Team Darzler from BSc first year. Here we go.
morning to one and all present here. Myself Ria. Today, me and my group are here to present a skit before you on the topic greenhouse effect. In our skit, Sadhna will be playing the role of atmosphere, sun and plains, Shravan as space and carbon dioxide, Spurti as the first heat ray and methane, and Ranjani as second heat ray, nitrogen oxide, cars and industrial gases, and me as your narrator. So let us know what is a greenhouse effect, how does it work, and how the gases affect the atmosphere.
Hello everyone, I am Sun. When my rays reaches the Earth's atmosphere, some of it gets reflected back into space and rest of them are absorbed and re-radiated by greenhouse gases.
Without the sun, people won't be able to live on the earth. But too much heat of the sun isn't good for us. The earth sends some of the heat to the space, and this helps to keep the earth from getting too hot. Now, heat rays travel into the space.
I am heat. I go back into a space.
I am space and some of the heat comes back to me.
Most of the earth's heat is trapped by the atmosphere, like the rooftop of the greenhouse. The atmosphere keeps most of the heat from going out into the space.
Hello everyone, I am atmosphere and I keep most of the heat from going back into the space.
From the second heat. The most of the heat and the stay in the atmosphere and warm it.
The atmosphere contains substance called gases. Some of the gases also trap the heat from the sun because these gases cause the greenhouse effect. They are called greenhouse gases.
Primary greenhouse gas is responsible for about 3/4 of emission. It lingers in the atmosphere for thousands of years. In 2018, the carbon dioxide reached 411 parts per million in Hawaii, Mauna Loa, atmospheric baseline observatories, the highest monthly average ever recorded. Carbon dioxide emissions are mainly come from burning of organic materials like coals, woods, oils and solid waste.
And heat. Methane is a main component of natural gas and it is releases from landfills, natural gas, petroleum industries and in agriculture, especially from digestive system of grazing animals. The mole of methane does not stay in the atmosphere as long as mole of carbon dioxide is above 12 years. But at least 84 times is more potent over 2 decades. It accounts above 60% all over greenhouse gas emission. Nitrogen oxide occupy a relatively small share of global greenhouse gases emission, about 6%, but it is 264 times more powerful than the carbon dioxide over 20 years. And its lifetime in the atmosphere exceeds a century, according to the IPCC. Agriculture and the livelihoods, including the fertilizer, manure, and burning of agricultural residues, along with the burning fuels, are the biggest source of nitrogen oxide gases. Fluorinated gases such as hydrocarbons, fluorocarbons, sulfur hexafluoride, and nitrogen trifluoride, and have a trapping potential thousands of times greater than CO2 and stay in the atmosphere for thousands to thousands of years. Accounting for about the 2% of all the emissions, they are used as refrigerants, solvents, and in the manufacturing sometimes occurring in the byproducts.
Some of the gases are produced by human activities such as driving and flying.
I am the car. We are the car, we make the greenhouse gases. Our personal vehicle are the major cause of the greenhouse warming collectively. Cars and the trucks account for the nearly 1/15 of all US emissions, emitting around 24 pounds of the carbon dioxide and other global warming gases for every gallon of gases. About 5 points comes from the extraction, production and delivery of the fuel, while the great bulk of the heat trapping emission, more than 90 pounds per gallon, comes right off the car pipe.
Hey guys, I'm blind and I do too. Emission from domestic aviation alone have increased 17% since 1990 to account for 9% of greenhouse gas emission from the US transportation sector. Flights departing from airport in the United States and its territory are responsible for almost one quarter of global passenger transport related carbon emissions, the majority of which come from domestic flights.
We make greenhouse gases by using fuels such as gasoline. Fuels are important. We need fuels to drive cars, fly planes, and heat our homes. But when we use fuels, gases go into the air and add to the greenhouse effect. The greenhouse gases effect helps to warm the earth. Thank you.
Mm, Thank you for such a nice role. So you have played a very nice role and showed how the greenhouse effect, like how the greenhouse global warming causes and the greenhouse effect occurs. Thank you for such an informative skit. Now let's move on to the another part, that is like now Bhavana, Harish and Kavita and Rakshita from team Eravithans are going to give a talk on the myth of science. Over to you guys.
Good morning, one and all. I'm going to talk about myths of science. Introduction to myth of science. A scientific myth is a myth about science or a myth or factoid that is commonly thought to be scientific. Scientific discoveries are often presented in a mythological way, with a theory being represented as dramatic flash of insight by a heroic individual rather than as the result of sustained experiments and reasoning. Myths about science. Science is always done with the scientific method. Science always done with an experiment. If we use the scientific method, we always get the right answer. Scientific information is proven by and never changes. Over to Bhavana.
Defining myth and science. The term myth is defined in widely different ways. Myth, the most narrow definition, treat as false narratives about the origin of the world and of humans. Science, It is the pursuit and application of knowledge and understanding of the natural and social world follow as a systematic methodology based on evidence. So, relation between myth and science. Just as myth, science attempt to create a coherent underlying worldview, which clearly not possible at almost any state of knowledge. Over to Arish.
Conclusion. The future of the study of the myth asks that the theories of the 20th century didn't challenge the supremacy of the science. So why we bother to accommodate the myths with science? Why not simply accept the 19th century view and dispense with myth in favor of science? The 20th century answers was restrictions of myth to either literal explanation or a symbolic description or physical events, fails to account for array of other functions and meaning that myths harbor. Thank you.
That was a nice presentation from all of you. Thank you very much for sharing such an wonderful topics. Thank you. Now let's move to next one. Now we have Divya Verma from MSc Microbiology to present about the wind of change. Over to you, Divya Verma.
Yes, so I'm going to present the winds of change role of bacteriophage in eliminating the antibiotic resistant bacteria. Good morning, everyone. It's long and interesting day. I wish your patience and attention for a few minutes. So here I'm going to present this topic, a role of bacteriophage in the elimination of antibiotic resistant bacteria. For many decades, the patients are behind iron curtain were denied access to some of the best antibiotics developed. Why is this so? Why the best antibiotics comes in this list? Why nowadays the third generation like ceftriaxone, cefotaxime, and fourth generation like cefepime are failing in their action? To make do some to make some interesting fact, the European countries are investing heavily in the use of bacteriophages. So the changing the way of medication and transforming into new treatment method. In that case, I'm exactly talking about the bacteriophages. The phages which have a specific action into a specific strain. In recent studies, the protection of phage from bacterial endonucleases is important. The bacteriophage should not affect the enzyme of immune system. So in here, the phages in this a virus are afraid. Yes, the people are afraid of viruses. So whether this treatment uh, should be play an important role in the future, it's very important. Phages, by contrast, kill just one species or strain. To find whether a bacteriophage for bacterial target is relatively easy or not. The the most challenging fact here is the phages should penetrate the eukaryotic cells and also do not affect its immune system. Yeah, it's very important. In this case, the phages kinetics action should be faster than the immune system, the way the immune system responses. I mean to say the phage, the phages kinetic, the action of phage should be at faster rate than the immune system respond. The main purpose of this topic is that the bacteriophage on antibiotic resistant bacteria show how consequently it shows the effect on immune response. Yes, there are many complications, there where the action is very complex, and also how it act on the immune system is also very important. The effect on human health, whether the human is consumable or whether it leads to a death.
These are the important factor which is very important to look on it. In order to overcome this, the exact treatment should exist. So the exact treatment, we can't say the exact treatment, but yeah, we are close to the treatment, that is bacteriophage, which cannot which only affect the particular bacteria, and it do not affect the immune system. It do not affect the normal flora of the body. Their antibiotics like third generation, fourth generation, they are broader. That is, the treatment by antibiotics is diverse. It's not specific. Antibiotics can kill out normal flora. Specifically, the broad spectrum of antibiotics kill many types of bacteria. So, here I presented some methodology through which we can at least achieve some goal to treat a resistant bacteria. So here are some phage, enrichment of bacteriophage, some phage assay by isolation and enumeration. We can isolate the bacteriophage from sewage by filtration, release by the release of viruses by carrying out some laboratory method. The filtration of sewage sample, enrichment of bacteriophage, and the release of virus from the cell. Then the detection of phage in the enriched lysate. This in that case, the in the log phase, the culture should be in the log phase on the host nutrient agar plate, and it is incubated for 24 hours. So after incubation, the zone of clearance are observed. Lysate should be preserved in the refrigerator at -4 degrees Celsius. So these are some procedure which are very helpful in the isolation of phages. Yeah, but the question may arises in your mind: can bacteria resist to phage? Yes, of course, bacteria can resist to phage. In that case, we can go for molecular engineering method. In that case, the phage engineer use a DNA editing system. The system called CRISPR to kill antibiotic resistant bacteria or phage resistant bacteria. In this case, the phages are injected into bacteria with DNA, and the microbial machinery called transcription will transcribe that DNA into RNA. If the gene in that bacteria is, if the gene, the bacteria, the resistant gene in that bacteria matches that RNA sequence, which is produced transcriptionally, then that enzyme called Cas9 will cuts up the cell's DNA, that is bacterial cell DNA, which leads to the killing of bacterial cells. So the next is, here are some problems and some findings related to the and conclusions. To identify a phage with therapeutic characteristics, whether the problem is whether the challenge is big here, where to identify a phage with their characteristics. Yes, it is very important. In that case, the phage should be injected in the bacterial cells. It can be identified by using specific probes like gene expert machines. It is an instrumental machine which is very specific to the gene, that is a transcribed RNA, which is very specific to the RNA. If this is appropriate, then we can analyze, we can go close to that. That's, yes, one characteristics, we can achieve at least to one characteristics. Bacteriophage do not have ability to spread throughout the membrane. Yes, in that case, we can allow the bacteriophage to spread through the membrane by using carrier, which do not act on immune response at a faster rate. Like the carrier can be the binding of lipids and avidin. If the lipids and avidin, the avidin and the lipid binds and act as a carrier on delivering the phage on the infectious site, specifically on pleural lining. On pleural lining, on the infection site is pleural lining. In that case, penetration, the phages penetrate, infect the cells by endocytosis, which found within early endosomes. After penetration, the phage will destroy all intracellular pathogen. This combination, like avidin, lipid, and cellulose, this combination can normalize the immune response. So the next problem is, can phage remain in body as long lasting? If the phage is specific to the host, yes, then it can remain in longer period of time. But it is very important that the phage should be specific. The phage should be specific to the host. Each passage is complex. How should we maintain that passage? For maintaining that passage, it is very important to clone the host population in each passage. The host population should be maintained, should be cloned in each serials of passage. So the conclusion here is the potential insight of evolution to improve the phage therapy against that pathogen is, we are very close to that, to kill that bacterial infection, to kill the most prominent infection at a specific site. Depending on the needs, phage sample could either be obtained from reference stocks or engineered through molecular techniques. Yes, for the generation of specific phages, the molecular techniques or the stock phages are important. But the most challenging here is phage use in this time is lapsed between obtaining the bacterial sample for the production. Specifically, the time is very important from the adaptation of phage, from the isolation of phage to the action of phage at the site of infection. So the main question is, humans are lacking with adenosine deaminase. Do phage therapy work in this case? In that case, the engineered phage is important. Adenosine deaminase is an inherited disease of immune system. ADA and that adenosine deaminase, an enzyme which involved in purine metabolism, which is essential for lymphocyte development. Yes, in that case, the phages are in that case the phages play an important role by treating an infection, because the modification is basically, it is a genetic modification in human, it is a genetic modification which is found, which have been occurred by the imbalance of the gut flora. Due to imbalance of the gut flora, the metabolites are secreted, which directly acts on the genes, and the modification occur. That modification is basically by the action of the normal flora. So in that case, that normal flora imbalances due to the bacterial infection, the competition of pathogenic organisms, and the normal flora. So pathogenic organisms compete then the normal flora, and the action here is gut flora imbalances, and it secretes the toxic product which has effects on gene. So here phage therapy will a challenging. In this case, the phage therapy will uh, directly affect the pathogenic organisms. So that pathogenic organism should be should have should inhibit the effects, the inhibition of the effects, so that the normal flora act on it, and it will stabilize the immune system response. These are the references through which I have went through for this topic. Any questions?
Any questions to Divya Verma? If there is no questions, then we'll move on. Thank you for sharing such an wonderful information with us. It was very informative. Thank you so much. Now, here we have Ajit, Aishwarya, Akhilesh, and Geeta from first year from the group The Black Hole to present a small skit on chemistry quiz through online.
Good morning, students.
Morning, ma'am.
Morning, ma'am.
Morning, ma'am. First, all of you mention your names in the chat box, students. Attendance is compulsory.
Okay, ma'am.
Yes, ma'am.
So, as you all know, today I'm going to conduct the chemistry quiz. So, I hope all of you are prepared well.
Yeah, ma'am.
Yes, sir. So, my first question is, what is the chemical name of CaCO3, students?
Yes, the answer, ma'am. Ma'am, CaCO3, ma'am.
Oh my god, it is not CaCO3, dear. It is calcium carbonate. Did you got it?
Yes, ma'am.
So my next question is, what is the chemical formula for water?
HIJKLMNO.
Oh, from where did you get that answer?
HIJKLMNO. H2O, right?
Oh my god, what an answer!
Enemies ahead! Let's go!
Akhilesh, what are you doing there? See, children, I'm so much worried about you all. Students, so I will continue now. My next question is, why hydrogen, carbon and oxygen belongs to the formal group? Can you all answer me?
Because they wear suits and ties, ma'am.
Yeah, ma'am.
Yes, ma'am.
Oh my god, Jit, what are you doing there? Jit, can you please stop that song and dance?
Sorry, ma'am.
So now I will ask the question for you only. Tell, what is the unit of resistivity? Whom is the right answer? Okay. Then what is the unit of power?
What, ma'am?
What? It's a right answer. See, students, all the students must be like him, Hajit. Jit.
Ma'am, thank you, ma'am. Actually, yesterday I was studying a lot, ma'am. So I can answer today's question, ma'am.
Bro, enemies ahead. Enemies ahead. Ma'am, still two only quiz now. Please wait.
Geeta, what are you doing, dear? What is that in your mouth? You're very obedient student, no?
Yeah, ma'am.
A few moments later.
Thank you, ma'am.
Yes, ma'am.
Yes, ma'am.
That was a very fun playing skit, and it was like I thought like what actually happening now in online. Thank you a lot for giving such a wonderful performance, and you thought like even science can be like made in a funny way. Thank you, guys. There is also an announcement that at the end of this meeting, we'll be announcing the result of Freshers Day. We'll be also announcing the Mr. Fresher and Miss Fresher in the end of this meeting. Please stay tuned. Now, let's move to the next part. Now we have Shalini and Kshitij from BSc second year to talk on interesting facts on science. Over to you both, guys.
Good morning, everyone. I am Satish.
And I'm Shalini.
We are here to amaze you with some curious or interesting facts of science. So get ready to be amazed with some extraordinary facts that anyone has never heard about them before. So let's not wait anymore and let us explore the facts together. Fact number one: bananas are radioactive. That how it is bananas are really radioactive? I will tell you because bananas contain potassium in it, and since potassium has a property of decaying, so that it makes potassium slightly radioactive. And you may be worried about that it can cause radiation poisoning to us by eating lots of bananas, but not to worry because you need to eat 10 millions of bananas at once to die because of radiation poisoning. So it's not possible at all. So you can eat bananas; it is not harmful. Fact number two: spider silk is stronger than steel. You know that steel is a hard element or it is strong as well, but not stronger than spider silk. How is this possible? Yeah? So I'll tell you because spider silk is light and flexible as well. So it may even be used for the soldiers one day to protect them from bullets and harmful things. And however, it is used to make products such as protective wear for the military and even for the surgical stitches. So this is how spider silk is stronger than steel. Fact number three: the human stomach can dissolve a razor blade. So you may be thinking that how our stomach can dissolve this thickened or sharp razor blade. So the human body is more capable than you think. Our stomach can dissolve this razor blade because hydrochloric acid is present in our stomach, which is a strong acid and having a pH of about 1 to 2 that can fully dissolve this razor blade. And also in the studies, scientists have found that the thickened bag of single edge blade is dissolved after the 2 hours of immersion in the stomach acid, which is HCl. But please don't try this. This is just a fact that you got to know now because it can be harmful for you also. Fact number four: only two letters which don't appear in the periodic table. So all of you know that periodic table and you have also studied it. So can anybody tell me which two letters are not in the periodic table? By seeing this periodic table, you can tell me. I'll give you 15 seconds to find it out. Which two letters are not there? Anyone can make guesses which two letters are not there?
Z is not there.
Z.
Z. Z is
Z.
there only. Anyone? Other?
J.
Yes, J and one more letter.
Satish, you only reveal the answer.
Yeah, okay. Okay, I'll only tell that the letters which don't appear in the periodic table are J, which was correct, and also the Q don't appear in this periodic table. So you can double check it also after this meeting. So fact number five: the smell after it rains. So how many people over here love the smell after rain? Because I do. But has anyone imagined what would the reason behind it? And what is the name of that smell that comes after the raining? So the particular name of the smell is called as petrichor. So this smell, petrichor, comes from an oil plants. The oil plants which decays on ground, and when it rains, this oil gets combined with a byproduct, which is a type of bacteria which is specially present on the dry ground, and it produces a smell called as petrichor. The next is fact number six: dreaming in black and white. So you may also have got an idea about this fact by seeing these images. Also, you can also tell on the basis of data, people would dream in black and white before the invention of the color TV. So in that time, people used to get dream in black and white color, and on an average, after the invention of color TV, person dreams in color compared to the previous data. So this shows that how our brain is done some adaptable changes of various kinds, And it's very interesting to see that how our brain is more susceptible to media nowadays. So let's see some more interesting facts by Shalini.
Thank you, Kshitij. So let's move on with the next fact. That is fact number 7. So webtastic. So web is such a nature's beautiful creation where we just knew only it as in some like useless dust accumulated in the corner of the houses, right? But no, these webs are very useful component which may act as an antiseptic and antifungal properties, which contain these properties. And these were used by Greece and Rome, Rome physicians to make bandages. So and also, these European medicines also use these cobwebs to to use it on wounds and cuts. And they were also found out that they speeded up the healing and reduced bleeding, due to which as due to the presence of rich vitamin K, this showed some of these properties, and also it showed few antifibrinolytic properties. So fact number 8, the color you see in the pitch of darkness has a name, guys. So as like Kshitij told, the smell of the rain after it rains also has a name. The same way, when you close your eyes, okay, when you close your eyes in a dark room and then open your eyes, and you see some shade of gray or black in a dark room, right? So that has a name. So that particular name of that darkness is called Eigengrau. So this is a particular shade of intrinsic gray, okay, where this is where it is observed when there is no light. So this is the particular shade of gray where people see when there is no light. So fact number 9, you can eat garlic through your feet. It's weird, right? So but it's somewhat true, because you can try it out yourself. Like if you rub a piece of garlic, okay, to in your sole of your feet, and then after 30 minutes you can taste, like you can feel the taste and also the smell in your mouth. It's all because of the organic compound called allicin, which is present in the garlic, guys. As each and every nature's component has an organic component in it. So allicin is also such a organic compound in this garlic, which is particularly formed when garlic is like chopped or like destructed, destroyed. So when we rub, obviously the garlic is destroyed. So and this allicin has such a power where to penetrate to the cell membranes, and also it travels to the bloodstream and reaches to the taste buds. So that is how you taste it. So that's the reason why, like people who love or like to eat garlic, like do not only have a bad odor in their mouths but also from their entire body, because this particular smell also dissolves in the blood. So fact number 10, dragonflies create a heart with their tails while mating. This is an perfect fact where you can see such a beautiful creation by the nature. So, as you can see in the image, this dragonflies undergo mating as the normal process, where it is only to symbolize, their particular change or transformation or many more. But these do not only do perform those only to symbolize those and all, like the previous said words, but also they show a true embody love in them, in them themselves. So as I told, they even create a combined heart, so which as you can see, it's nature's beautiful creation. So this is a fact where a relationship of any bonding is extremely executed. So fact number 11, vampire bat saliva keeps away blood from clotting. In general, whenever a wound is occurred and the blood oozes out from any of the body part or the prey, so the blood tends to clot, right? So the same way, but these vampires feed on their prey on their blood, right? So if it gets clotted, how can they feed? So that is why the nature has given a boon to these vampire bats, that a protein in their saliva act as an anticoagulant, so that when they feed on the prey, the blood can flow freely. So, and another fun fact relating to this is, like a protein, right? As I told, which acts as an anticoagulant, was named as Dracula, Draculin. Dracula, spooky character of vampire in many Irish or English novel or movies. So fact number 12, the higher your IQ is, the more interesting your dreams are. So this is one of the most interesting fact, because scientists claim that, based on the studies, people who are very intellectual and also with a higher IQ had a very really captivating dreams, and also brighter and more long lasting dreams when compared to the people with lower IQ. But don't get upset, like don't demotivate yourself that if you have a bad dream you have an lower IQ. No, but you can like increase your IQ. And also don't get upset like if you can't remember any dreams which you saw the last night, because even I do also do not remember any some of my dreams which I saw in the last night, because on an average a dream lasts only on a couple of seconds. So you can think that facts are like, why did we choose this topic, right? So the reason we choose this topic is because like to exhibit on this platform is that learning new thing is amazing, right, as these facts do. So by the end of this presentation we would like to quote that the more that you read, the more things you will know, the more that you learn, the more places you'll go. So let us always keep learning new to grow more. Thank you.
Thank to you both guys, like for giving such an wonderful facts. It was so interesting. Like from this I hope everyone would like to would be curious and would be very interested to more about the science. Thank you for giving such an wonderful facts. Now let's move. We have Ajit from BSc first sem to show his miming talent through a short video. That was such an entertaining act, Ajit. Thank you so much for sharing such an good talent with us. Thank you very much. Now let's call upon Harshita, Nilesh, Pallavi, Parvati, and Sneha to present their presentation on aminosynthesis.
Good morning, everyone. I'm Pallavi. Welcome to Science Forum 2021. And we are going to present our seminar on the topic amniocentesis. Our group members are Harshita, Pallavi, Parvati, Nilesh, and Sneha. The contents that we have included in our presentation are amniocentesis, who is candidate for amniocentesis, detection through an amniocentesis, complications of amniocentesis, and results. And finally, conclusion. What exactly is amniocentesis? A procedure is performed usually in the beginning of pregnancy to detect chromosomal abnormalities in the fetus, usually done when a woman is between 16 and 22 weeks pregnant. During this procedure, a small sample amount of amniotic fluid is taken from the amniotic sac surrounding the unborn baby, and its DNA is examined for genetic abnormalities. In order to understand amniocentesis, we have to understand some of the parts of the body and how they work. Who is the candidate for amniocentesis? Any pregnant woman who at a due date will be 40 years of age or older, any woman whose combi test results are high risk, blood tests or fetal ultrasound. Now I would like to welcome my friend Parvati to continue.
What can be detected through an amniocentesis? Amniocentesis detects chromosomes abnormality, neutral tube defects, and genetic disorders. Nearly all chromosomes disorders include Down syndrome as well as sex chromosome. Amniocentesis does not detect all birth defects, but it can be used to detect the following conditions like sickle cell diseases, spina bifida. Complications of amniocentesis. 1% of risk of miscarriage and pain from the procedure, and also increases risk of clubfoot. Injury from needle and also infections due to the amniocentesis. Now the results from amniocentesis. After amniocentesis, the sample of amniotic fluid is sent to a lab for analysis. Results usually take 10 days to 3 weeks depending on the lab. It takes that long to get result because the cells have to grow in the lab in order to analyze them. In the lab, genetic and chemical tests are done. For genetic tests, certain chromosomes and genes are analyzed. Now the conclusions. Amniocentesis may cause club foot in your baby. Amniocentesis is a safe procedure that can provide helpful information about the health of a fetus. It may be offered to a woman who are at higher risk of having a baby with a genetic disease. It is the patient's decision whether the benefits of the procedure outweigh the risk. Now here are some reference for the amniocentesis. Thank you.
Thank you guys for such a nice presentation. It was very informative. I hope everyone would have would have learned something from this presentation. Now we'll move on to the next one. Now we have a topic to present, that is antibiotic resistant by Sahana, Tanisha, Tejashwini, Venkatesh and Yogita. Over to you guys.
Hello. Hello. Good morning to everyone. Our topic is antibiotic resistance. First, about science. Science is about a whole lot more than that, and to sum it up, we believe that science is a way of helping brain grow in finding new knowledge and helps us depict our curiosity of how the world develops and works today. Science is important because it has helped from the world that we live in today. And our topic is antibiotic resistance. Our group members are me, Tanisha, Sahana, Tejashwini, and Venkatesh. In antibiotic, hello. In the antibiotic resistance, what we are learning of history of resistance, antibiotic resistant crisis, why resistance is concentrate, mechanism of resistance, generation of resistance, antibiotic resistance, second and third generation of antibiotic resistance, producing enzymes that inactives the antibiotics, and new antibiotic development, efflux pumps, conclusion and references. First, what is antibiotic resistance? Means it is the ability to bacteria and other microorganisms to withstand to antibiotics and inhibit the antibiotic resistance. In 1945, Fleming himself warned of the danger of resistance. According to him, it is not a difficult to make microbe resistant to penicillin in the laboratory to by exposing them to concentrations not sufficient to kill them, and the same thing has occasionally happened in the body. And next slide, the history of the resistance. My friend Sahana will take over. Sahana, over to you, dear.
Good morning to one and all. This is Sahana. I'm here to explain history of resistance. Throughout history, there has been a continual battle between human and mutate of microorganisms that cause infection and disease. Penicillin was successful in controlling bacterial infections among World War II soldiers. However, shortly thereafter, penicillin resistance became a sustained clinical problem. In response, new beta-lactam antibacteria were discovered, developed, and deployed, restoring confidence. Figure in next slide show the various of crisis in antibiotic resistance. Here we can observe the various of antibiotic resistance. Why resistance is concentrate? Resistance organism led to treatment failure, increased mortality. Resistance bacteria can be undetected. Added burden on healthcare costs. Tends to return to pre-antibiotic era. Selection pressure. Now, I would hand over to my friend Tejaswini.
Hello everyone. Now I'll let you know about the mechanism of resistance. The abilities of bacterial organisms to utilize the various strategies to restrict antimicrobial compounds are all genetically encoded. Intrinsic resistance is the type of resistance which is naturally coded and expressed of intrinsic resistance. It's a natural resistance of anaerobes to aminoglycosides and gram-negative bacteria against vancomycin. Acquired resistance changes in bacterial genome through mutation or horizontal gene acquitization, on the other hand, may consequently lead to the change in the nature of proteins expressed by the organisms. Such change may lead to the alteration in the structural and functional features of the bacteria involved, which may result in the changes leading to resistance against the particular antibiotic. In fact, several different mechanisms may work together to confer resistance to a single antimicrobial agent. Now let's know about the generation of antibiotics. Generation term comes only in case beta E of penicillin and cephalosporins beta-lactam antibiotics and depending on their action on the cell wall of gram-positive and gram-negative bacteria. They are classified by using terms broad-spectrum and narrow-spectrum antibiotics. Here, broad-spectrum means the antibiotics which are effective for large and narrow spectrum is effective only for limited range of antibiotics. Let's know about first generation antibiotics. First generation antibiotics have a narrow spectrum of clinical use, good for common gram-positive bacteria that cause ear and throat infections, venereal disease of gonorrhea and syphilis. A very high number of drugs in this group are resistant to organisms that produce penicillinase. Now let's know about second and third generation antibiotics. Second generation antibiotics have extended or intermediate spectrum of clinical use. Some are gram positive and some may be gram negative. Not very effective against penicillinase producing organisms. However, their optimum activity is against gram negative bacteria, which is not resistant to penicillinase producing organisms. Fourth generation antibiotics are extended spectrum antibiotics. They are not resistant to beta-lactamase producing microorganisms. Fifth generation antibiotics are extended spectrum antibiotics. They are cephalosporin, ceftriaxone, which causes pneumonia, skin, and soft tissue infection. Cefetobiprole, which is methicillin resistant Staphylococcus aureus. Now I'll hand over to Venkatesh.
Hello, good morning everyone. Myself Venkatesh. Now we can know about by producing enzymes that inactivate antibiotics, inactivation of beta-lactam antibiotics, Staphylococcus aureus, Neisseria gonorrhea, and Haemophilus influenzae produce B-lactamase, which cleaves beta-lactamase ring. Inactivation of chlorophenicol. This is inactivated by chlorophenicol acetyltransferase. And gram-negative enzyme present continuously, hence higher resistance bacteria enzyme is inducible. And inactivation of aminoglycosides. It is inactivated by acetyl, phosphor, and adenyl transferase present in gram-positive and gram-negative. And we can know about new antibiotic development. Only 15 antibiotics of 167 under development had a new mechanism of action with the potential new combat of multi-drug resistance. Lack of incentive for the companies to develop antibiotics. We can observe in the table. Now I'm handing to my friend Tanisha.
Good morning, everyone. So,
I'll introduce myself as Tanisha Chakraborty. By new antibiotic development, that is lack of incentive for companies to develop antibiotics. Only 15 antibiotics of 167 under development had a new mechanism of action with the potential new combat of multi-drug resistance. And this is the graph of drying up antibiotics approved by US Food and Drug Administration. And efflux pumps, it is a cytoplasmic membrane transport protein, major mechanism for resistance in tetracyclines. Some gram-negative bacteria inhibit the plasmid-mediated synthesis of protein channels, which obstructs the influx of hydrophilic penicillin, example ampicillin, ATP binding acids, major facilitator, superfamily, multidrug and drug-resistant transports, small multidrug resistant transports, resistance nodulation division. Lastly, we would conclude this topic of antibiotic resistance by saying that the importance and value of antibiotics cannot be overestimated. We are totally dependent on them for the treatment of infectious diseases. The best practice can be that doctors and healthcare centers should provide their patients places that are resistance free by taking strict measures in infection control and antibiotic use. There is no perfect antibiotic, and once the most appropriate uses of any new compound are identified, it is essential that prescription be restricted to those uses. And so everyone should be thinking, so why we chose this topic? It is because antibiotics are one of mankind's most important discoveries. They allow us to survive serious bacterial infections. These are some references which we took from completing this topic of antibiotic resistance. Lastly, thanks to everyone, especially thank you to our respected chairperson and teachers for your precious time and my dear friends for listening to us so patiently. Thank you.
Thank you to you guys. Like you have said it was very nice presentation and you have said very informative things. It was very informative. Thank you, you guys. Like next, we have another skit from Manoj, Nagarjun, Nandita, Pooja, or from the group Expedition Science from first year BSc. On the topic of this skit is COVID-19 and 20.
Hey people, I'm COVID 19. I came from China to India. I love traveling and I travel through air. So I'll move to person by cough, sneeze, and fever. So let me play with you guys and I'm here to destroy all your peace.
Hi everyone, this is Pooja. I am here to advise you people who are all suffering from the mild symptoms of COVID 19 in this pandemic condition. Don't be panic or worried if you got a positive result in your COVID 19 test. We are here to help you out of it. You can also manage it by home itself by making your home quarantine in an isolated room. Our Government of India and the World Health Organization had given many precautionary measures. You can follow that also. And we doctors are here to help you out of it and by giving our best treatments and make you better as fast as possible. So let us see what are all the precautionary measures we must have to follow. Clean your hands frequently using your hand wash or any soaps, it's your wish. And you can also wear the face mask and gloves whenever you have to go outside. Use a warm water always for your drinking purpose. If you want to cough or sneeze, use your bent elbow instead of your hands. Don't touch your sense organs like eyes, nose, and mouth. Limited social gatherings are required in this pandemic condition, so don't involve in the crowded entertainments and avoid a closer contact with the person who is in the sick. Use disinfectants and sanitizers by keeping your surface and the objects which are using frequently by us. And the vaccines are arriving soonly and we doctors are working on it, and let us all fight against the COVID-19.
God, what should I do now? Why this vaccine came? Oh my God. Now all corona went, COVID went, fear went. Oh my God. Let me introduce myself. This is mixed version of COVID-19. COVID-19 and COVID-20. More powerful than COVID-19. Can you see the blue dots? This is my protein wall. I can travel through it more faster than COVID-19. It can travel 25% faster and I can travel 70% faster, and be ready to bear me. Sure, we'll have a fun time.
I am Nagarjuna and I am here to tell you some precautionary measures how to avoid the COVID-19 and COVID-20. You should wash your hands regularly. You should maintain the social distance in the society, sanitizing the belongings and the hands of you, and wearing the mask whenever you go out, and avoiding the street food, whatever it available in the streets or bus stands. Please do follow, friends. Save your health. I thank one and all who are present here, and I especially thank Principal Anuradha ma'am for her valuable time, and also I thank Dr. Netra ma'am for the guidance and the encouragement for us. Thank you once again. Thank you.
Swati, your audio is muted.
Yeah, yeah. Thank you, guys. It was very nice. Like you shared what actually happened in this pandemic. It was very informative. Thank you for you made it interesting way. Thank you, you guys. Now let's move on to the next one. Now we have Manoj is from first year BSc to present his posters on COVID-19 and 20. Over to you, Manoj.
Hello everyone, myself Manoj from BSc Genetics first year. So this is my poster presentation for fighting COVID-19 and COVID-20. So wash your hand. Yes. So first step is washing hands daily, frequently washing your hands so that the virus, whichever on our hand, it will destroy. Then second is maintaining physical distance. Then third is avoiding touching eyes, that is sensitive parts of our faces like eyes, nose, and mouth. Then wearing mask whenever we go out, that avoids us from preventing COVID-19, and we can fight COVID-19 and upcoming COVID-20, that is a new version of COVID-19. Thank you.
Thank you, Manoj. It was nice poster presentation. In a short way, you have presented it very well. Now let's move to the next part. Like we have Mugdha Hegde from first year to speak on plant meat. Now, Mugdha, you can talk about your topic.
Um, Hello, good afternoon, everybody. My name is Mugdha Hegde, BVOC first year.
Today, my topic is plant meat. That is, actually, it is a plant-based meat. Plant-based meat, an alternative for animal-based meat. So today, I wish to like I'm gonna cover the points like introduction to plant meat and the reasons why we should bring or adapt plant-based meat instead of animal meat, and the types of plant-based meat and ingredients used in it, the advantages and disadvantages of using plant meat, and later the conclusion. So, coming to the introduction part. So, first, actually, meat means the actual definition is it's actually the flesh of any animal, like birds or any animal. But since we don't know the exact word, we don't know the official name of for this plant-based meat. Let's call it as meat itself. So yes, coming to the introduction, plant-based meats are those which are made from extracted plant proteins or whey proteins. For that, spices are added and binding ingredients to help them to bind. So those ingredients are mixed. The main component, the main ingredient is the protein. The plants which have the plants and the plant products which have more proteins are used for this, and these types of meats are designed to mimic meat in every way. Like it might be to it like it is a replacement for the original meat. It might be in the in the matter of taste, texture, color, all these things, even in the appearance. So they are trying, and it is designed so to exactly to make it look like the original meat. And this plant based meat sector has been growing and becoming a new topic of interest for food scientists and technologists. So, actually, this is the main reason why I chose this topic, since even I am a food, just a minute, even I am like a food industry student. Yes, it is becoming a new new topic of interest for all food scientists and technologists. Like, what are the advantages and disadvantages? It's still in the discussion, like how people will receive it and all those things. Yes, it is. It has already come to the market. Like, many people have accepted it. Okay. The next point is this meat comes in the form of burger patties, hot dog, that is used in the like the sausages. Okay, and the ground meat, nuggets, even in the form of seafood like shrimp and fish. In all forms, the meat, it might be of any animal. They're trying to mimic all kind of meat. Okay, and this meat is produced by a process called extrusion. So we BVOC students, we have come across this extrusion. Actually, extrusion, it is used to make pastas and all these, the dry, the pastas and all spaghetti you may have heard. For that, basically extrusion is used. Here, extrusion is used to bind the plant base and all those spices and ingredients, what are the things which are used to prepare it. And it is also been considered as food for future. Like, people are saying also in the many websites, or people are commenting that this may become a permanent replacement for the animal original meat. Okay, next slide. So, what are the reasons to bring plant-based meat? Why we should replace the animal meat by plant-based meat is the ingredients used in it. So, since we know the animal meat, it has very less advantages, in case of health, it has very less advantage and more disadvantage. So the ingredients used in this are all plant based itself. The proteins, the all the nutrient contents what we get from plant, we can get in this plant based meat. So the first point is the ingredients used in it, and the way people have accepted it. So as I told, those who had produced the factories which had produced plant based meat, they were worried like how people will receive, will it fail or what. But no, people have received it in a quite good way. Okay, it's still gaining popularity. So yeah, the next point is to prevent the environment harm caused by animal based meat. So you may think, you may get a question that what environment harm can the by consuming meat, what environment can get affected. So the reason how environment is harmed is for example, the in a farm, let's consider a people who are rearing pigs for their meat, or the poultry farm and all. They will make a piece of land. For that they should go through the deforestation. They should undergo deforestation. For that, trees will be cut and all those things. And the crops which the like pigs and sheep, all those things, all those animals need, those kind of crops are grown for that. So precious water, soil, trees are destroyed from this. And moreover, the animals untreated waste, it will pollute the rivers and the streams. Okay, and comparative health benefits, as I told, by consuming by having animal based meat, it is said that it has more cholesterol and chances of high rate of cancer and heart diseases are more. So and plant based protein, like plant based meat, we have nowhere heard that from plants we get cholesterol or we get heart diseases and all. So in that way, comparatively, the health benefits are more in this plant meat. And yeah, major meat producers are investing on this plant-based meat, plant-based alternatives, and etc. Okay, next point. Next slide. So coming to the types of plant-based meat, there are primarily two main types. That is, one is whole muscle meat, and another one is restructured meat. As the name itself suggests, whole muscle meat means it is not cut or something like that. Its shape will be the same. So to mimic that, like muscle meat may be like steak, chicken breast, etc., like that. Non-vegetarian people may be knowing it. Okay, to mimic that. And then another one is restructured meat, ground beef, meatballs, sausages, nuggets, and etc. So these two are the main things. Yeah, next slide. So what are the ingredients used? Okay, basically there are some common ingredients, and there are some main ingredients also. The common ingredients are like grains and legumes. You can see the what are legumes and the grains used. And some main ingredients are soy and its products. You may have heard of soya bean or soya chunks. If anyone have eaten soya chunks, it will taste a little bit like meat itself, like not taste but the fibrous thing. And seaweed and algae, mycoprotein, myco, that is fungi, mushrooms, I can say. And pulses, hemp protein. Hemp is actually a plant. From that protein is extracted. Using that protein powder, it is made. And the third picture, the lentils and some seeds such as pumpkin seeds, watermelon seeds, etc., are also used. So what are the advantages if we come across? Since animal-based meat have both cholesterol and saturated fat mainly, and plant-based meat have no cholesterol and comparatively less saturated fat. It also have saturated fat, but comparatively less. So plant-based meat is preferable, and reduces animal suffer. Like it will help those people who are animal lover, and like they don't want, they want don't want to kill animals but still want to eat meat. So those people can adopt or can start using these plant-based meat. So for those kind of people, it is more helpful, I can say. And people can live healthier life. As I told, the risks from animal based meat is more compared to the plant based meat. Diseases that spread through animals when we eat their meat can be prevented. So there are many diseases. For example, the present thing, the bird flu. So at least if we can't, if permanently, if we can't start using plant-based meat, we can at least make use of these plant-based meat at these times when diseases or some other climatic conditions like these times. And using plant-based meat is eco-friendly. Yes, next slide, please. So there are not only advantages but also disadvantages. Like some of the meat substitutes to look realistic may be containing excess food coloring, texture, additives. So to make it look like realistic, to look exactly like the original meat, to make people tempt, they will add more colorings and textual additives. And it is high in sodium. It has more sodium content in it. And you're using plant-based meat may cause changes in the food chain, like population will increase. The like if we stop eating fish, okay, their population will increase. And if we stop eating goat, sheep, or like other meat and all, their population will increase, and it may affect the food chain. It is my personal like opinion, my personal point. And compromise with taste and so on. So we have to compromise with taste. It is said that it won't give the exact taste what the original meat gives. And yeah, coming to the conclusion part, plant-based meat represent the kind of change the world needs at a time when climate catastrophe is wreaking havoc, and many people are suffering negative health effects from consuming too much meat. Like yeah, as I told, the climate condition, or at this time of when diseases from certain animals are spreading, we alternative for that, we can actually adapt these, actually start using plant based meat. And always consumers preference stand first. What we consumers, what we customers demand, that will always stands for first. Our preferences stand first. If we demand, if we oppose the plant based meat, it won't like it can't be marketed. Or if we prefer the plant based meat, it will of course our preferences will be respected first. And anything which is consumed in a limit is always good. So yeah, yeah, thank you for one and all for giving your precious time. Have a nice day. Thank you.
Thank you, Mugdha, for giving such an informative presentation. And you presented it very well. Thank you for such a nice presentation. Now, like, let's move on to the next one. Now, Lisa would be giving a speech, like talking about the space. I know space means it's a wonderful topic. Now let's, Lisa, it's over to you. You can speak about it.
Stars, moon, constellations, beautiful to look at, hiding many secrets. Things like space and universe has always fascinated me in a way nothing else did. It's so vast that it's called a space with pitch blackness of nothingness. Good morning. Oh, actually, good afternoon. I'm Lisha from BWork first year, and today I would like to give you a little bit of a sneak peek on what's beyond our solar system. But before getting into the interstellar space, we need to know about the first two man-made spacecraft built and launched in 1970s to leave our solar system. Next slide. The Voyager 1 and 2. No other spacecrafts have ever traveled that far. Now, these twin Voyagers 1 and 2 are exploring where nothing from the Earth has ever flown before. In August 2012, Voyager 1 made the historic entry into the interstellar space, whereas Voyager 2 entered the interstellar space on November 5, 2018. Continuing their more than 40-year-old journey since 1977, they are still sending scientific information through the Deep Space Network yet. Next. Moving on to the closest star to our solar system, Proxima Centauri. Now, this star comes under a triple planetary system. The system consists of three stars, Alpha Centauri A, Alpha Centauri B, and Proxima Centauri. Proxima Centauri is the dimmest star out of the three and is around 4.2 light years away from the sun. This is the image of Alpha Centauri A and B from the sun. Coming to exoplanets. Now, all the planets outside our solar system orbiting other stars are called exoplanets. These planets revolve around the star, just like how the planets in the solar system revolve around the sun. The planet revolving around the star Proxima Centauri that I just mentioned is called Proxima Centauri b, and it is the closest exoplanet to the sun. Next. Black holes. Now, this is one of the most mysterious and strangest things ever in the universe. Black hole is made up of a dense collection of matter. Now, this matter is packed into a very, very, very small space or volume. And because of this, it has a very strong magnetic and gravitational force. It's so dense that not even light can pass through it. Sagittarius A is known yet as the closest black hole to us. The first ever black hole image was taken with the help of Event Horizon Telescope last year, that was 2019. 8 such telescopes were actually installed all at the different places around the world to take this image of M87 black hole. It was a really difficult task, but we made it come true. Next is the Milky Way galaxy, our galaxy. Now, this galaxy consists of billions of stars and billions of planets, but it's not the only galaxy in this vast universe. We have hundreds and millions of more galaxies of different sizes and different shapes. Andromeda galaxy is the closest to our home Milky Way galaxy. And these are the different type of galaxies that you can see, like irregular, spiral, peculiar, etc. And mind that the Milky Way galaxy comes under a spiral galaxy. Okay, next. Nebula. Nebula is, you can say that it's an interstellar cloud or cluster of dust and different types of ionized gases. It's a cluster of all these different materials. and it comes together to form a nebula. Now, Orion Nebula is the brightest nebula in the sky or in the space. Now, it is also said that nebula are the star forming regions. That is, it's a place where stars are born or formed. There is so much more to know, yet we know so little. But it's okay, since the curiosity is never ending about everything that lies ahead. Thank you.
Thank you, Lisha, for sharing such a wonderful and amazing facts about space. Yeah, it's a wonderful topic if you take about space, and there is a lot more to know. Thank you, Lisha, for letting us know many things today. Now, it's come to an end. Now, before concluding this meeting, I would like to call upon Manju Bhargavi ma'am to announce the Miss Fresher and Mr. Fresher and the results of Freshers Day. Ma'am, it's now over to you.
Good morning, dear students,
Good
and
morning,
my dear colleague friends. It's my pleasure to announce results for Mr. Fresh and Miss Fresher 2021. And on behalf of our beloved principal and my dear colleague friends, I am announcing the results. Here, the category of talent search, they made two category actually before announcing Mr. and Fresher. The talent search results as follows. The first place for a talent search that goes to Mr. Sai Krishna, CND person. And second place goes to Miss Kamai, BSc CND person. And the third place goes to Miss Anaga, BSc CND person. And consolation prize is there for Miss Nandita, BSc person. And this is the time to announce Mr. Fresher and Miss Fresher, UG 2021. And the Mr. Fresher of UG goes to Mr. Manoj, BSc person. Mr. Manoj, BSc person is the Mr. Fresher. And Miss Fresher, UG 2021 goes to Miss Nandita, BSc person. I congratulate all the winners and as well as the organizers of Freshers Party 2021. Congratulations everyone, and thank you so much Netra ma'am and Sunita ma'am for giving this opportunity. Thank you Principal ma'am. Thank you one and all. Have a nice day. Thank you.
Thank you, ma'am. Thank you for making the announcement. Thank you, ma'am. It was a great opportunity to have you here, ma'am.
Yes, and on behalf of everyone, I congratulate all the talent search winners and also Mr. and Mrs. Fresher. And whoever hasn't got, don't get upset, guys. There is more and more functions or competitions ahead where you can express yourself. So I thank everyone who has been a part of this meeting and also has made it successful. And also, if before ending this meeting, if any teachers are interested to speak few words, you're free to speak, ma'am.
Yeah, this is Dr. Shanta. Yeah, dear students, I just want to congratulate all of you for your eager participation and presence for in the science forum. I was so surprised to see your efforts in preparing the slides and a skit and for the poster presentations. I really love your imagination. We teachers, actually, we have to learn a lot from the young minds, I hope. And as Anuradha madam said, actually, this is the platform you can exchange your views and thoughts among yourselves and ourselves as a science, technology, and many more environmental issues. I hope this platform definitely inculcate your inquisitiveness. I request all the students from all the departments, please think of many more innovative and purposeful programs in the future. And also, I want to congratulate to Mr. and Miss Freshers and all the winners. Love you all. Thank you.
Thank you, ma'am. It's because of all you, like all the great teachers like you people, ma'am, making us so motivated and like making us more interactive and become more friendly with you people, ma'am. Thank you, ma'am. And Swati, I think you can head over to the vote of thanks.
Yeah. Now, like it's time to conclude this meeting. Thanks to one and all. Without you, this event would not have been successful. On behalf of PIMS, I would like to thank Shri Narayanappa Sir, the Managing Trustee and the Guest of Honor, Padma Shri Institute Group of Sciences. Shri Satish Sir, the Chairman of Padma Shri Group of Institute, and Professor Sanoj Sir, the Director of Padma Shri Group of Institute. I would also thank our beloved Principal Ma'am, the Guest of Honor, Dr. Anuradha, the Principal of Padma Shri Institute of Management and Science. I would also thank Sunita Ma'am, Netra Ma'am, and Akshay Sir, the organizing team. If it was because of you who organized this, it was successful and it was good. So I would like everyone, thanks to all the faculty member and the student for the cooperation. Thank you one and all.